because for the past few years, that's what I've been doing. I've been trying to characterize what's wrong or what's not wrong in these tau knockout cells. And I try to look at different phenotypes, treating them with amyloid beta and see whether there's any sort of rescue. Of course, the next step is to focus deeper into the mechanism. For example, we know there's an impairment in neural outgrowth. We want to know why. The lack of tau protein, why is it the case that there's an impairment in neural outgrowth? So we are looking at several different possible mechanisms, but of course, I can only speculate because I don't really have the data yet. So that's deeper into the mechanism. That's what we are looking at. And actually, the tau knockout neurons project is only half of my PhD. The other half is actually we are trying to use sporadic AD patient-derived stem cells to model the disease somehow, comparing with the clinical phenotypes. So apart from these tau knockout cells that we made in the lab, we also have a panel of patient-derived stem cells that we can try to, whatever we see in the tau knockout cells, we can try to look at certain phenotypes in these human stem cells from the patients. And this is a rather big project because in order to model sporadic disease, they don't have any mutations. So you need a pretty large number of stem cells, which can take up a lot of time. Yeah, so that's the next step, actually already currently undergoing